Mitosis and Meiosis by Senopoulos
and Semendinger.
The cell cycle is the process of the cell dividing, consisting of interphase and mitosis. Interphase. This process consists of G1, S, and G2 phases. This is where the cell spends the majority of its time during the cell cycle. G1 is when the cell grows and the volume of the cytoplasm increases. During the S phase, or synthesis, the cell duplicates its DNA. During the G2 phase, the chromatin is duplicated and the cell resumes growth in the preparation for mitosis. Mitosis. This process is important for cell growth, the reproduction of new cells, and the maintenance and repair of worn-out cells. Prophase. In this phase, the chromatin condenses, becoming visible, combining into structures called chromosomes. There are two identical copies of each chromosome in the cell, sister chromatids, attached at the middle by centromeres. The nucleolus disappears, nuclear membrane breaks down, and the centrioles move to the opposite sides. Metaphase. In this phase, spindle fibers form. Chromosomes align in the middle of the cell along the metaphase plate. Anaphase. In this phase, centromeres of the chromosomes separate and move to opposite poles of the cell. Since each sister chromatid has a centromere, it is now called a chromosome, and thus there are twice as many chromosomes in the cell compared to the beginning. Telophase. During this phase, the chromosomes move towards the ends of the cells and form into daughter nuclei. The nuclear membrane begins to reform and the chromosomes vanish. Poof! Cytokinesis. During this phase, the cytoplasm is divided. A cleavage furrow will form, separating the two daughter nuclei and forming two completely new cells. Each cell now has half the original material of the original cell. At the end of mitosis, the results of chromosomes is 92, with 46 in each of the two new cells formed. This is the same amount of chromosomes as the cell began with, so by the end it effectively doubled. Furthermore, each of the new cells contains half of the material that was from the original cell. The composition is therefore half of what it was when it began the process.
The purpose of meiosis is to divide the cells that will be used in the process of sexual reproduction in eukaryotic cells. In humans, this process of meiosis is used to make egg and sperm cells. Meiosis 1. The purpose of this is to separate homologous chromosomes. Interphase 1. During this process, chromosomes duplicate, much like they do in mitosis. Prophase 1. The longest phase of meiosis, homologous chromosomes come together to share segments of DNA. These newly intermingled chromosomes are now known as tetrads. Metaphase 1. In this process, the newly intermingled tetrads line up on the metaphase plate. Anaphase 1. In this process, the pairs of homologous chromosomes split as a result of shortening kinetochore microtubules and begin moving towards opposite poles of the cells. Telophase 1. As the cleavage furrow forms and the cell separates into two parts, two haploid cells form. Both of these new cells now have a new nuclear membrane surrounding them, and the microtubules that were formed in the process earlier begin to disappear. Meiosis 2. After telophase 1, the cells may enter one final process called meiosis 2. The same process occurs in meiosis 2 that occurred in meiosis 1. The only difference here is that instead of separating homologous chromosomes, meiosis 2 focuses on separating the result of meiosis 1, which is sister chromatids. This results in 4 haploid daughter cells being formed, each containing a single chromosome. The difference between mitosis and meiosis is that the result of mitosis holds 46 chromosomes, while the result of meiosis holds only 23. The reason meiosis only holds 23 is because it is used in the reproduction of organisms where the two cells will eventually combine to make a whole cell.